Assalamualaikum and salam sejahtera to our lecturer Dr. Nurul Hikmah binti Harun. We are from Group 3. We'll present about the tumor immunology. Firstly, as for introduction, let me introduce the audience what is tumor. Generally, the tumor is abnormal mass of tissue that forms when cells grow and divide excessively. The cell may be benign or malignant. As for your information, benign tumor cannot spread into the nearby tissue or other parts of the body. However, malignant tumor can spread into other parts of the body through blood and lymph system. Secondly, what is tumor antigen? Tumor antigens are proteins, glycoproteins, glycolipids or carbohydrates that express on the surface of the tumor cells. Below is the animation that shows the normal antigen and tumor antigen on the surface of cell. Next, move on how our body reacts to tumor. I will explain in general. So, basically, antibodies will bind to the antigen on the tumor cells, triggering and promoting cell destruction by other immune system cells. And the last part in this slide is the causes of tumor. There are so many causes of tumor. Below is a few of example. The tumor may be developed due to gene mutation, radiation exposure, environmental toxins, and chemical exposure such as benzene.
For the first question, we will discuss about types of tumor antigen. Initially, tumor antigen were generally being classified into two types, which are tumor-specific antigen and tumor-associated antigen based on their pattern of expression. However, this type of classification is imperfect because many antigen thought to be tumor-specific antigen turn out to be expressed on some normal cell as well. In modern classification of tumor antigen and the latest classification, tumor antigen are being classified based on their molecular structure and sources. Listed in the slide are types of tumor antigen that being classified based on their molecular structure and sources, such as oncofatal antigen, oncoviral antigen, overexpressed or accumulated antigen, cancer testis antigen, CT9-CT10 antigen, lineage-restricted antigen, mutated antigen, post-translationally altered antigen, and idiotypic antigen. Next, in this slide, I will explain two examples of tumor antigen, which are mutated antigen and oncofatal antigen. Mutated antigen is a tumor antigen in which protein produced by a tumor cell that has an abnormal structure due to mutation. Mutation in the relevant gene cause abnormal protein to be generated. Tumor are caused by mutation in proto-oncogene and tumor suppressors which result in abnormal protein production. These abnormal proteins are referred to tumor-specific antigen. The abnormal product of P53 and CDK4 gene are examples of tumor-specific antigen. Other genes unrelated to tumor formation, on the other hand, may lead to the formation of abnormal protein and known as tumor-associated antigen. Next is oncofatal antigen. Oncofatal antigen are another important type of tumor antigens. Examples of oncofatal antigen include alpha-fetoprotein and carcinoembryonic antigen. These proteins are generally created during the early stage of embryonic development and then disappear after the immune system has fully grown. As a result, self-tolerance to certain antigen does not develop. However, oncofatal antigen may appear due to the repression of genes that were only expressed early in life.
Next is immune mechanism of tumor rejection. In here, we want to discuss how the immune system is. In immune system, there are conceived of innate and adaptive immune system which play a critical role in suppressing tumor growth. The innate immune system act rapidly as the first line defense. However, when this system is unable to destroy the pathogen completely, the adaptive immune system will activate. In innate immune system, the immune cell that inhibit tumor cell are natural killer cell, which is NK cell, dendritic cell, or known as DC cell or antigen presenting cell, and macrophage. While in adaptive immune system, the immune cell that involve are T cell and B cell. Natural killer cell act by destroy tumor in non-specific fashion, where it will recognize infected and stressed cell and respond by killing this cell. NK cell also will attach to the cell that lack class 1 MHC antigen, which this action will release cytotoxic perforin and grant enzyme from secretory granule. In here, tumor antigen will stimulate the dendritic cell, which is act primarily as antigen-presenting cell. These cells are capable of presenting processed peptide from various antigen, initiating and modulating the adaptive immune response by activating both T and B lymphocyte. The dendritic cell will present the tumor antigen by MHC1 to T cytotoxic cell, which is CD8, and by MCH2 to T helper cell, which is CD4, activating cell. Then, for macrophage, it act by display a variety of cell surface receptors that enable them to recognize and engulf pathogen through the opsonization process. Lastly, in cell mediated, which is carried out by T cell, are consist of T cytotoxic and T helper cell. These cytotoxic effects the cell cause tumor cell death by fast ligand binding to the FAR receptor on the tumor cell that trigger caspase activation and apoptotic cell death, and also by formation of post-performing perforin complex that release perforin and granenzyme from granular tumor cell. T helper cell 1 will directly kill tumor cell by release of cytokine that activate the death receptor on the tumor cell surface. Meanwhile, for T helper cell 2, it will activate B cell that produce plasma cell that produce antibodies. The antibody recognition of antigen on tumor cell will result in tumor cell death by ADCC, opsonization, and complement activation. Through all these immune cell mechanisms, it will inhibit the tumor growth.
Hi everyone, I am GUNASEKARAN and I'll be presenting about the induction of lymphocyte apoptosis. It is the loss of immunological competence, which means the immune system is not sensitive enough to kill the tumor cells. Okay, in this case, the cancerous cells will express the FAS ligand instead of the FAS receptor. And by this way, it hijacks the immune system. Now, why do I say that? Because in a normal immune response against tumor cell, it is the T cell, the lymphocyte, that will express the FAS ligand to kill the tumor cell. But in this case, the opposite happens. And this FAS ligand is a type 2 transmembrane protein. It is a membrane-bound cell surface protein that belongs to the tumor necrosis factor superfamily. The TNF is mainly involved in cell proliferation, regulation of the immune system, and apoptosis. So the main idea is the binding of the FAS receptor on the lymphocytes to the FAS ligand on the tumor cell will cause the apoptosis, the programmed cell death on the lymphocyte instead of the tumor cell. And that is the main idea. And how does it happen? It happens through two separate FAS ligand based mechanism and through a classic negative feedback loop. And the two type of apoptosis occurs is autocrine apoptosis and paracrine apoptosis. In this slide, I'm going to explain how it works out. Okay, let's focus on only diagram A first. We can see the tumor cell here, it expresses the FAS ligand. So it is symbolized as FAS-L positive. This FAS ligand on this tumor cell will go bind to the FAS receptor on this T cell, which usually doesn't happen in a normal immune response. So basically the tumor cell is cheating the immune system, it's hijacking, and this is how hijacking occurs. It binds, it causes a signal transduction. It transduces a signal known as the caspase activation. The caspase activation is the activation of proteolytic enzymes to kill the cell, enzymes that causes the cell death, the programmed cell death. So that is how the death of the T cell occurs instead of the tumor cell. Okay, and in diagram B, we can see that tumor cell that does not express the FAS ligand is labeled as FAS-L negative. So in this case, the tumor antigen on this tumor cell is presented by the major histocompatibility complex class 1 protein known as the MHC class 1 shortly. And this MHC class 1 protein that presents the tumor antigen on this tumor cell in diagram B binds to the T cell receptor and causes a formation of the complex. When this occurs, the interaction occurs, it causes the activation of this example, the cytotoxic T cell. It causes the activation of this T cell. And when this T cell activated, it causes an expression of the FAS ligand on the T cell itself in an autocrine fashion. Autocrine means the production and secretion of extracellular mediator through signal transduction. So an autocrine signaling occurs in which a new FAS ligand is formed on this T cell which bind to the FAS receptor on the same T cell. Because we know the T cell have FAS receptor, and by doing this they interact with each other on the same cell and they die, they suicide. Labeled by this small arrow through caspase activation and leading to apoptosis. So that is the first mechanism of the FAS ligand mechanism to kill the lymphocyte. The second FAS ligand mechanism is when the T cell is being activated by the complex MHC1 TCR complex, the FAS ligand on this T cell will go bind to the FAS receptor on another T cell. So instead of killing itself only, it kills another T cell together. It fratricide, and it's symbolized by this asterisk sign, and it die by caspase activation and apoptosis. So this is how lymphocyte apoptosis occurs.
As for the mechanisms of tumor evasion, There are three mechanisms, which includes low immunogenicity, antigenic modulation, and tumor-induced immune suppression. So, for the first mechanism, which is the low immunogenicity, anti-tumor activities are strongly depends on the effectiveness of tumor-associated antigen TAA presentation. TAAs are presented and processed by dendritic cells to induce the effector T cell activation. Downregulation of MHC1 limits the effectiveness of anti-tumor activities. It is achieved by mutation of the beta-2M gene, a component of the MHC1 molecule, which can cause a complete absence of MHC1 expression, and it is also achieved by the mutation in TAP transporter or components in immunoproteasome such as latent membrane protein LMP2 and LMP7. Next, for antigenic modulation, it is an ability of an antibody to cross-link of two antigen molecules which triggers cellular signals. It accelerates endocytosis and degradation of those molecules. Endocytosis causes cells absorb external material by engulfing it with the cell membrane and cause degradation. And for the tumor-induced privilege site, it is caused by the factors known as Fas ligand, which is secreted by tumor cells, create a physical barrier to the immune system. It is a type 2 transmembrane protein that belongs to the tumor necrosis factor family. Its binding with its receptor induces apoptosis.
Next, immunological approaches for cancer therapy. First, chemotherapy. This is a procedure to inhibit cell proliferation and tumor multiplication by avoiding invasion and metastasis. But this results in toxic effect of chemotherapy due to the effect on normal cell as well. Inhibition of tumor growth can take place at several levels within the cell and its environment. Second, stem cell transplantation. Stem cell transplant are procedures that restore blood-forming stem cells in people who have had TRs destroyed by the high doses of chemotherapy or radiation therapy that are used to treat certain cancers. Third approach is immune checkpoint inhibitors. This is a drug that blocks immune checkpoint. These checkpoints are a normal part of the immune system and keep immune responses from being too strong. By blocking them, this drug allows immune cells to respond more strongly to cancer. The mechanism of action of this drug are shown below. Approaches for cancer therapy is by using monoclonal antibodies. Monoclonal antibodies are immune system proteins created in the lab that are designed to bind to specific targets on cancer cells. Some monoclonal antibodies mark cancer cells for better seen and destroyed by the immune system. Next, by using T-cell transfer therapy. This is a treatment that boosts the natural ability of the T-cell to fight cancers. In this treatment, immune cells are taken from the tumor. Those that are most active against cancer are selected or charged in the lab to better attack the cancer cell, grown in large batches, and put back into the body through a needle in a vein. The last immunological approaches is immune system modulators, which enhance the body's immune response against cancer. And some of these agents affect specific part of the immune system, whereas others affect the immune system in a more general way.
The last part is conclusion. As a summary, tumor can be made up of benign or cancerous cells. These cells have tumor antigen on their surface. The immune system sees the antigen as foreign substance and recruit an immune response against them. This helps the body fight cancer. On the other hand, certain type of cancer can be identified by this tumor antigen, which act as a biomarker. As for example, alpha-fetoprotein and carcinoembryonic antigen can be used as tumor markers for liver and colon cancer respectively. More importantly, tumor antigen also involved in cancer therapy as tumor antigen vaccine. Here are the list of references. That's all from us. Thank you.